Let's talk about energy. So we're going to start with cellular respiration, move on to photosynthesis, and if we have time, we will be discussing enzymes, which are heavily involved in each of the two earlier ideas. So we're going to loop them all together if we've got time for it, and then we'll practice. So to remind you, the first part of cellular respiration, and this occurs in all living organisms, is glycolysis. And all, yes, in some way, shape, or form. Sorry, I'm going to say yes. Yes, glucose will be broken down. It requires some ATP. It takes a little energy to break this down. It takes a little energy to break anything down. And then that kind of charges it. It breaks into two parts, G3P, glyceraldehyde 3-phosphate, and this, which are really the same thing, just different versions. That's going to happen 2 times. And so for each glucose, you're going to get an NADH, which is our friend that is going to go to the electron transport chain if we have oxygen, and we get out 2 ATP per G3P. So again, 2 ATP in, this happens twice, so there are 4 ATP out and 2 NADHs and a pyruvate, which we're going to see later as well. Now, if there is no oxygen, this isn't going to go anywhere. This NADH can't be used up by the electron transport chain. And we need NAD+ in order to make glycolysis happen. So we'll do anaerobic respiration, otherwise known as fermentation. There are 2 flavors. The first of which is what we do, what lots and lots of animals do, lots of organisms do, where glucose will generate some ATP at this stage, and then you get the pyruvates. But instead of those pyruvates going into the Krebs cycle and then generating things for the electron transport chain, they're going to get converted into lactate or lactic acid, which is where you get sore muscles from. Now, that NADH, instead of going to the electron transport chain, is going to get converted back to NAD+, which you need in order to run glycolysis in order to get these ATP out. So that's lactic acid. Now, if you were, say, a yeast or certain types of prokaryotes, you could do alcohol fermentation where ATP, again, is generated. And then that pyruvate gets converted into acetaldehyde and then ethanol. It generates CO2, which is why we could make beer and bread. And again, that NADH gets converted back to NAD+, which is needed for the glycolysis part of fermentation. So that's the first part, either without oxygen or aerobic respiration that requires oxygen. And then the pyruvate is going to get converted into acetyl-CoA, which feeds into the Krebs cycle or the citric acid cycle or the TCA cycle, all names for the same thing. But this again only happens if you have oxygen, because these electron carriers, these buses full of electrons, are going to go to the ETC where they get used. So overall, acetyl-CoA is going to combine with oxaloacetate to make citric acid, citrate, or citric acid, depending on the pH, which is the name of the cycle. It generates NADHs and CO2s. So you get carbon dioxide here. This is where the carbon dioxide byproducts of cellular respiration come from in aerobic. That's going to, you're going to lose a couple of carbons there, and you're left with this 4-carbon molecule. You're going to get a GTP, which gets converted into an ATP. So this is some substrate level phosphorylation that we get just like we had it with the glycolysis. And then you get FADH2, which is a, don't forget that H2 here, some people forget it, which is a big bus full of electrons, and NADH, again, which is a big bus full of electrons. Really, more than anything, understanding these intermediates and what goes off. You don't need to know all the little things in between here. Knowing oxaloacetate, knowing citrate are good, and they're going to be helpful, but all the other things in between aren't super necessary. But remember, going from NAD plus to NADH, that's reduction. Going from NADH to NAD plus, the more positive, that is oxidation. So remember, you can think of reduction as reducing the positive charge. You see, we go from positive to no positive. We add an H. We add H's, and all of these are evidence that this is a reduction occurring here. And then electron transport chain. These big buses full of electrons, NADH and FADH2, are going to donate their electrons to the electron transport chain, which is the energy that drives this. Remember, this is a type of active transport because this is also going to give H pluses when it breaks down and gets oxidized. The NADH and the FADH2 get oxidized into NAD plus and FAD. Those H pluses are going to move into the intermembrane space, and this intermembrane space is going to allow basically storing energy that gets harnessed to generate ATP. We're going against the concentration gradient. So we are adding from a low area of low H plus to high H plus. So it does require energy, which is in the form of these electrons. And as it goes through this very famous enzyme, the ATP synthase, it's going to generate ATP, which you need for all sorts of things. I think that's the last thing I wanted to say before asking you guys if you had any questions. Are there any questions at this point? I went through a lot in a short period of time, but I'll see, are there any questions? Okay, since I'm not seeing any yet, I'm going to move on to photosynthesis. Now, this is our friend, the plant cell, and it has chloroplasts, which are the organelles responsible for photosynthesis. It's also got all the other fun organelles in the cell, including the mitochondria. Don't forget that mitochondria is also in plant cells. Some people forget. I'm sure you won't. And I wouldn't worry about amyloplast. That's a storage center for like carbohydrates. But ribosome, cell membrane, cell wall, these are all going to be in there. A big central vacuole that stores water is going to be unique to plant cells, something to remember. And also, you're not going to see the centrosomes, the centrioles, in the same way like you do in animal cells. But the chloroplast, it has very important parts to it, parts that are important to take note of. It's got the thylakoid membrane. That is actually going to be the site of the light-dependent reactions, which we're going to talk about more, and they're going to be in stacks called granum. Remember, just while we're here, there's an inner membrane, an intermembrane space, and an outer membrane. Remember that outer membrane is evidence that this is used to be a prokaryote that was enveloped by a cell, because it's that primitive membrane. Anyway, the thylakoids is where the light-dependent reactions are going to occur, and then the stroma, that space outside of the thylakoids, is going to be where the light-independent reactions occur. People don't say dark reactions anymore because they're occurring during light times. But yes, not in the thylakoids. That space is called the stroma within a chloroplast. And there's some chloroplast DNA, again, good evidence for endosymbiosis. So let's talk about the light-dependent reactions, which are occurring again on that thylakoid membrane. Inside of the thylakoid is going to be what's called the thylakoid lumen. That's the little inside of the pancake. And then outside, as I said, is the stroma. So light is going to come. It's going to hit the photosystems, which are, basically going to work best at different wavelengths. But what happens is water gets oxidized. It's going to lose its H2s, which are going to help build up this gradient. The electrons are going to start there and then get excited by light. So that light excites the electrons, kind of charges the electrons, so that they can move down their down this electron transport chain. It's going to pull H pluses from low concentration into this high concentration space. Again, this provides the energy. These high energy electrons provides the energy to pull those in. Again, the electrons, which are in a low energy state, get excited again, so that they can actually help provide the energy necessary for reducing NADP plus into NADPH. Don't forget there's a P in there for photosynthesis. So pH, and that's this again, just like in the electron transport chain of cellular respiration, is going to be a big wall, a big dam of H pluses that want to flow through ATP synthase to create and generate ATP, which is going to be used in the Calvin cycle, or the light independent reactions on the next stage of photosynthesis. Don't forget that this is again, this ATP is being generated by ATP synthase, and it's NADPH that is going to move on to the Calvin cycle. Speaking of which, let's talk about it. So again, don't pay too much attention to, there's a bunch of intermediates in here, and Mr. W talked about it. You don't need to know the big details, but you do need to know this overall idea. Rubisco is the enzyme that is the most abundant in on Earth. It's very prevalent because producers are the most prevalent life form on Earth, and these producers are going to, because they need to support the rest of life, these producers all have Rubisco that takes CO2 from the atmosphere and fixes it. It's the carbon fixation step, where it takes air CO2 and attaches it to other molecules that can be eventually used. So RuBP, don't forget, RuBP is the 5-carbon molecule that gets CO2 added to it. Rubisco is the enzyme. So this CO2 get added to RuBP. We have the 6-carbon molecule, which will degrade into 3 PGAs. They have ATP. They get phosphorylated, which kind of sets up the step for the reduction step that eventually gives us G3P. So NADPH is going to get oxidized into NADP plus. Again, you're losing an H. That's good evidence that it's gotten oxidized, and you will create 1 G3P. 5 are going to be recycled in order to create the necessary RuBP to keep the cycle going, but 1 leaves in order to create that G3P. And don't forget that you need 2 of these, because glucose is a 6-carbon molecule. So you need 2 G3Ps in order to make 1 glucose. And again, there's another ATP step, so you can see that it's going to add 3 more ATPs need to come in order to recharge the Ruby P. That is the light independent reactions. So I like this slide. It kind of ties the two together, and they really like asking questions where they are tied together. So if we look at photosynthesis, light comes in, charges those electrons. It's going to create ATP and NADPH, which are the electron carriers that feed in and power the Kelvin cycle. That's going to generate PGAL or G3P. They're the same thing. When you get glucose, other organic molecules, that is going to feed into respiration, which takes a little bit of ATP and NAD plus in order to power glycolysis. You get 4 ATPs, so again, you get a net of 2 ATP, and you get 2 charged NADHs in the reduced form. Those pyruvates, after they are converted into acetyl-CoA, are going to power the Krebs cycle. The Krebs cycle is going to produce NADHs and FADH2s that power the electron transport chain of respiration, which is going to generate, let's not forget H2O. I kind of glossed over that fact. I'll go back to it. That oxygen, the reason we can do aerobic respiration and the reason we need oxygen is because it's the final electron acceptor, and it gets reduced into water at the end of cellular respiration. So don't forget that. If you don't have oxygen, you can't do it because it can't accept the electrons at the end of the electron transport chain. But again, that is going to produce water. The oxygen from light-dependent reactions as a byproduct, when it gets water gets oxidized, is going to feed into the ETC and vice versa. Okay. Do I have any questions at this point? Let's see, what time are we at? Talked a lot, but I might have time for enzymes. Especially if there's no questions. So, okay. Okay, this is just another slide showing the interdependence of photosynthesis and cellular respiration. They really like asking questions that talk about this overall idea that H2O and CO2 feed into photosynthesis. The byproducts or the products of photosynthesis are glucose, C6H12O6, and oxygen, which feed into cellular respiration, which gives us the usable energy. This is a good figure too. Now let's talk enzymes. So anytime, almost every enzyme question that I've seen is they're going to talk about the fact that the structure of enzymes influences the function of the enzyme. So if there's an enzyme question and you can fit into the answer this statement, the structure affects the function, you're going to do well. They really like, and then back it up. They really like that. Readers really like that idea. It's a big idea for AP biology. And when I'm talking about that, I basically mean the structure of the enzyme, the actual active site, and even the allosteric sites, like where a non-competitive inhibitor would bind, are super important, because of the enzymes, which are proteins, they need to be a certain shape in order to catalyze a reaction. If they are not that shape, because they've been denatured, they are not going to catalyze that reaction. Just reminders about some inhibitors. If it's a competitive inhibitor, it's competing with the substrate. So the substrate can't bind to the active site, so it can't make the products that it's supposed to make. If it's a non-competitive inhibitor, sometimes the substrate can bind, sometimes it can't. Sometimes there is a conformational change in the enzyme because of the a non-competitive inhibitor binding there. But either way, the reaction is going to be blocked because of the inhibitor not allowing this product to be formed. So that's happened. Oftentimes it is that a non-competitor inhibitor gets in here and that makes the active site change shape, and then the substrate won't come in there too. Some things that affect the rate of enzyme activity. Big one's temperature, for sure. As the temperature increases, the enzyme activity will increase until up to an optimum, and then you get denaturation of that enzyme. And they really like asking about denaturation to really make sure you understand how that enzymes are proteins and proteins are affected by temperature. So if something gets too hot and you see a lowered activity, make sure you talk about how the enzyme is likely denatured, as evidenced by the decrease at reaction rate. There's an optimum temperature, just like there's an optimum pH. If you can actually, there is an inactive level where you increase the temperature and it's reversible. But if you get like a fever for too long, that's going to lead to denaturation of enzymes. pH is also super important depending on where the enzyme is and where its activity is, because most of the body is at 7.3 to 7.4. Most organisms are going to be at that pH. That's going to mean that most enzymes have an optimum at that level. But the stomach is very, very acidic. And so like a stomach enzyme like pepsin is going to be optimized at that. And if you get into just neutral pH, like water, it's not going to work. And then small intestine is alkaline, it's basic. So it is going to have an optimum as well at its specific pH. And then, I'm going to go through this quickly. Relative amounts of enzyme. If you have more enzyme, you're going to be able to increase the rate of reaction. If you have more substrate, you're going to also be able to increase the rate of reaction up till a point. Because it's like workers in factories. If you have 10 workers and you have tires, a certain level of tires, they're going to be able to make more cars. It's a car factory, right? But if you have 10 workers and you have 1 million tires, well, they're not going to be able to make 1 million cars. There's only a certain number of cars they can make in a given day, even if you have lots and lots of supplies. So you're limited in that case to enzymes in that case. So this would be like a substrate concentration rate of reaction graph. Okay, and then I guess I didn't hit it real hard, but this is just words that might come up. If you're building something, that's anabolism. If you were building proteins, that is building larger molecules from smaller molecules. Enzymes are responsible for it. That's why I'm including it here. And if they're an enzyme that's breaking something down, that's a catabolic reaction. Just the words, language that might help you in understanding something. I remember cats like to knock things down and break them. And if you're jacked up on anabolic steroids, you might have really big muscles that you've built up. There are lots of problems. I don't advocate you to take steroids unless it's medically recommended, but yes, that is a case. So I think that's the whole shebang. The big things that I wanted to go over, and I'm just at 1 o'clock right now, so I'll stop now and ask you guys if you have any questions for me. And if not, we'll get to practicing the FERC. That's FRQ, in case you're wondering. It's FRQ. All right. My wonderful, wonderful students, since I'm not seeing my chat box light up, I'm going to stop recording, and this is where we're going to start the FRQ.